Okay, so our next topic is the mass selection versus pure line selection. We have studied the different methods of selection in the plant breeding program, under which we have studied separately how we do the mass selection, and we have also studied the pure line selection and how these two methods are different from each other. Now, in this particular lecture, we will understand how the mass selection method is different from the pure line selection, and what are the other basic differences which are there throughout the process between mass selection and the pure line selection. So we are dealing with the paper number 2 and the chapter number 3, that is plant breeding biotechnology and the biostatistics, and we are here at the selection. Now consider this slide. This slide shows you the 7 basic differences between the mass selection and the pure line selection. Consider the first one. See, the pure line, the mass selection contains the mixture of pure lines, while on the other hand, pure lines shows the set of uniform characteristics. When we talk about the mass selection, from the population of a plant, we select the several plant which shows the specificity or say superiority for the some of the characteristics which are of human interest. Basically, if there are a total 100 plants in the field, we select the 10 plants which are showing the very good height, the very good growth, the very good grain size, or showing the very high disease resistance variety. So in and all, we are considering many factors in selecting the particular plant from the population, and hence it contains the mixture of pure lines. Okay, for example, you can consider the resistance to the diseases as a one pure line, then the higher size or bigger size of the grain as the second pure line, then it is showing the very good height which is called as the third line, pure line, right? In this way, many pure lines are involved in the mass selection. While on the other hand, when we talk about the pure line selection, it is only a set of uniform characteristics, because only the one plant is got selected from the population whose progeny is showing the superior characteristics, and hence from this one plant we will have only the uniform type of the characteristics. So in the mass selection we have the mixture of pure lines, while on the other hand in the pure lines we have the uniform set of characteristics. Then in the second, it usually involved self as well as the cross pollination. In a mass selection, we trace on both the types of the plants, that is self-pollinated plant as well as the cross-pollinated plants. But in the pure line selection, we only go for the self-pollination type of the pollination, because in a pure line we want to conserve the genes. Okay, we do not require any type of the variation among the genes which are selected earlier, and hence we only trace on the self-pollination. Whereas in the mass selection, you can go for the self pollination as well as for the cross pollination, because we doesn't care here for the variation. Okay, so that is why this brings us to the third point, that the genetic variations are present, because there is a self pollination as well as the cross pollination involved. In the cross pollination, the two types of the parents are involved to create the third species, and hence the genetic variation is largely found in the mass selection. While this type of the genetic variation is generally absent in the pure line selection, because at the beginning what we are selecting is only the one plant, and once we select this particular one plant which is showing the superior characteristics, we want to maintain the characteristic of that one plant only. Okay, this one plant should be there in order to conserve its all genetic material, and we compulsorily avoid the cross pollination so that there will not be any dilution of the genes which are already selected. Okay, so in the mass selection you have the presence of the genetic variation, while in the pure line you didn't find there is any genetic variation. Okay, now this is a particular mass selection method is the old method of selection. Okay, right from our ancestors we are doing this particular method of selection of the crop plants so that we can have the improvement, okay, in our agricultural field. But this pure line method is a newly developed method. Actually it is discovered in the 1903 only. So nearly 120 years ago we have the pure line selection, but this mass selection method is there from the ancient time. So mass selection method in the plant breeding is quite an old method, while the pure line selection method is quite a new method. Then the more variation and hence the more stability, if there is a more genetic variation in any organism, that organism tends to be very stable in an ecosystem because they shows the higher degree of adaptability. Hence in the mass selected plant, this mass selection method shows the higher degree of variation among the genetic material, and hence these plants are more towards the environment, and hence this shows the more stability. While on the other hand, these are in the pure line selection, you will have the very less variation, and hence they are less stable in the ecosystem or in the nature, because they do not have that much type of the genetic variation which can make them suitable for the any kind of environment. So on the one hand, in the mass selection, you will have the variety of genes presence, and hence they are quite stable in the ecosystem. While on the other hand, in the pure line selection, you will have the less variation and hence the less stability. Then the heterozygous variety is there in the mass selection method, because there is no uniformity in the genes. Okay, genes are variable, and hence we say these are the heterozygous varieties. While in the pure line selection, we have the homozygous varieties, because they were all showing the similar kinds of the genes. Okay, because they were coming from the one parent, because only the self pollination is involved. But in the mass selection, the cross pollination involved and the many plants are involved, and hence the heterozygous variety is created. Okay, then in the population of a plant got selected, while on the other hand, in the pure line, only the one plant is got selected for the progeny, which is showing the higher superiority in terms of the characters. Okay, so when we select the plant for the mass selection, we tend to select the group of plants which are showing the superiority in different characteristics, while in the pure line selection, we are only going to select the only one plant which is showing the one or two superior characters, and we want to conserve that particular characters within that plant. Hence we go for the self pollination type of the pollination. Okay, so these are the basic 7 types of the variation which are there between the mass selection and the pure line selection. You should remember them carefully, because UPSC can directly ask you the question that what is the difference between the mass selection and the pure line selection. All right.